Okay. Cool. So conference has been going pretty well so far. Yeah. Do you want to introduce yourself and what you do and what brought you to the Neural Interfaces conferences, I guess?
So my name is Jacob Robinson. I'm an assistant professor at Rice University in the electrical engineering department. My background's in nanotechnology devices, things that can be made tiny, scalable, and I also have a background in applied physics, so I think a lot about how energy is transduced in different ways. At some point, I decided that it would be interesting to see if we could use devices to try to hack into the brain of the peripheral nervous system to read and write activity. And so I started that during my postdoc. What brought me here is we've been working on thinking about what would be the next generation of neural interfaces. And we've been exploring the idea of using light as a way to maybe replace electrical interfaces. Part of this idea is that we have an eye toward the future where maybe through genetic manipulations we could create brain cells. We're not creating brain cells. The idea is that, with an eye towards the future, we can manipulate brain cells, have them produce genes that they don't normally produce. Mm-hmm. That will allow us to use light as a way to either control brain activity or to record brain activity.
So optogenetics, then,
exactly
right?
Right. Yeah. And specifically for the readout, we'll be thinking about things like calcium-sensitive imaging, voltage-sensitive imaging. So then the question is, all right, if this is going to be the reality, how then are we going to build the devices that are going to either deliver light to the nervous system or to collect light from the nervous system in a way that can be implanted and miniaturized and placed inside the body? Mm. And so the technology specifically that brought me here today is the idea of taking microscopes, integrating them onto a single silicon chip, and placing them on top of the brain or perhaps inside of the brain. And they would work a lot like maybe electrodes you've been familiar with, like Utah rays, but instead of actually, communicating with electrical signals, it would be communicating with light.
And what are the advantages of that?
The advantages of using light would be specificity, mostly. So both cell type specificity as well as spatial specificity. So when we think about electrical stimulation, it's difficult for us to control exactly the cells that are responding to that electrical stimulus. It's also hard for us to selectively stimulate cells of one type versus another type. But this is something that we have the ability to control by using genetic techniques. Use cell type specific promoters to say, okay, just excitatory cells, just inhibitory cells can be activated. From the readout standpoint, one of the challenges that we have for high density readout of neural activity is that there's a difficult source separation problem. So if I start listening to many neurons at the same time, how do I know if that particular signal is coming from one cell or its neighbor? We have ways of processing those signals to do spike sorting, and that works reasonably well, but we get something like one cell per electrode. One of the motivations for us to work in the optical domain is that if we can take images of brain activity, that means the source localization problem becomes much easier. Because you can say, okay, this is how the signal varies in time, but because we have spatial information, I can know exactly the source of those signals.
But wouldn't you still have like diffraction and stuff like this, and you wouldn't really know the depth, something like that? Like it might be maybe not like a sphere of like, okay, the neuron could be within the sphere, this is radius or something like this. It would be more of a cone, something like that.
Potentially, but I think one of the things that we're finding is that with advances that people are making with microscopy and image processing, we're doing a really good job of being able to solve the source localization problem in the presence of scattering and in the presence of diffraction. So we might be able to take an image that looks blurry and then with signal processing make that image look much sharper. And so some of this idea is coming from diverse fields in signal processing, compressed sensing, and we can apply those techniques to image processing for specifically identifying the locations of neurons that are active.
And so would it be, again, like one electrode, one neuron, or could one like microscope see multiple neurons, and would it be able to distinguish them as well?
Yeah. So I think the mapping that I would see for an optical interface would be at best 1 pixel per neuron.
Oh, okay.
But if we think about this, actually we're in great shape, right? Because we all have megapixel sensors in our pocket.
Yeah.
So if I were able to achieve this 1 neuron per pixel dream,
that's amazing.
Something I would have would be 1 million channels.
Yeah. Wow.
And so that's part of the motivation is that, for a variety of reasons, semiconductor manufacturing has pushed image sensors to the point where we have tiny pixels, we have millions of them on a chip. They've solved a lot of the amplification, multiplexing problems that we have for electrodes at a much greater scale. And so now the idea is, can we then design the algorithms as well as the optics that would be interfacing with those chips?
Literally tear apart a camera and be like, we're using this now.
Exactly.
And they spent billions of dollars, to have the fabs and everything to build it up. And research money would never allow you to do that. So might as well use something off the shelf. That's really interesting. But what about the data processing of this? I mean, already with 100 channels or 1,000 channels, something like that, the data is just so huge, and you need big, lots of processing power to sort through it, and then all this. But this sounds like a whole nother level. It's almost like so with the podcast I do audio editing and video editing, right?
Yeah.
And the video editing is a lot more power intensive, and it takes a lot more, significantly more processing power. I would say at least, even 100 times more. So is this something similar, or because already they pretty much have a server for one array?
Yeah.
Would this be 100 servers per one array, or what?
Yeah. You bring up some interesting points. I think the challenge maybe is not as daunting as it might seem at first blush. And that's because we already have a lot of techniques to compress video. So we might be sampling from a megapixel sensor, but there are a variety of ways we can press that down, particularly when our signals are sparse, either in space or in time. So that helps. But you're right, there is going to be some intensive post-processing that will have to happen. And this will likely happen kind of offline, at least in the early stages. So the model that we have is we collect data as fast as we can. And we can stream that data off of the sensor. Processing that data might take a while. But for basic science, this is not a big problem, because you would perform your experiment, you would collect your data, you would observe animal behavior. After the fact, you can then go back to your neural signals.
Chew terabytes and terabytes.
Chew through that.
What's after terabytes? Zettabyte?
Petabytes.
Petabytes, yeah. So, what kind of data sizes do you think? Would you expect? You haven't built it, right? Or what would you expect?
Yeah, so I guess maybe we can rewind a little bit, and I can tell you a little bit about what our technology is. So, starting with off-the-shelf sensors, we can then put a mask on top of that that's only a couple of 100 microns thick. What that does is it transforms the incident light in such a way that we can collect information about the angle of all of the rays that are coming in. It's not a lens.
Oh, it's not a lens?
No.
How's it different than a lens?
So a lens has this very specific point spread function, meaning that light that originates from the focal point will get focused down to a point on the opposite side. And we know that based on imaging with lenses, there are things like diffraction limits, and there's concepts like numerical aperture, the greatest angle that I can sample that light. What we do instead is we say, look, the challenge of actually creating or reconstructing an image can be solved computationally if I were to know the angle of all the rays that were coming in and their relative amplitudes. Now, so what that means is that I just have to sample the wavefront in such a way that I have enough information to reconstruct an image. So it's not forming an image on my sensor. If I were to actually look at what I capture, it would look nothing like an image. It just looks like a mess. It looks like noise. But because we know the transfer function that we're applying, we can apply the inverse of that transfer function. So you can think about this maybe from a signal processing standpoint, is that we're applying a transformation, then we're undoing that transformation to recover an estimate of the original scene. And now that estimate isn't a perfect reconstruction, but it's good enough for us to understand where light is coming from and the intensity of that light.
How imperfect is it? Like what kind of loss do you have?
It's hard to estimate what the losses are. I guess I don't have a good answer for you, and it also depends on the properties of the scene, how much light we have. What I can say is that we've been able to reconstruct green fluorescent protein. What can I say about that? There's really not a good metric for how good That's a really how can I answer that for you?
Just say maybe I don't know. It's hard to measure, right?
I would say it really depends on what you're looking at.
Mm-hmm.
I mean, I guess this is true for all of us, right?
Mm-hmm.
If you were to take your cell phone out and take a picture, and you were to look at that same thing, you'd say, well, how good is that picture compared to what I'm looking at?
Mm-hmm.
It depends what I'm looking at. It depends on how bright it is.
Yeah.
It depends on how far away your object
Exactly.
is. So it's very difficult for us to say how imperfect it is. So it depends on many different factors. What I think is important is that because we're not using a lens, we don't suffer from many of the trade-offs that we have to contend with when we work with lenses.
Mm-hmm.
So you do photography, I see stuff around here. So you're probably familiar with the idea that your depth of field is related to the aperture that you're working with. Right? So this is a property of a lens. Turns out that for microscopy, we have similar trade-offs. That trade-off usually is the field of view is related to the numerical aperture. So basically, I can collect a lot of light if I have a large magnification and therefore have collect light from a very small area. And this is a fundamental trade-off that we have with lenses. Now, if we say, okay, we're not using lenses, we're just going to take light, we're going to try and figure out what the angles and all the amplitudes and reconstruct that image, then I no longer have this trade-off between numerical aperture and field of view. So I can collect a lot of light, and I can collect that light from a very large area. And this makes sense because the idea is I'm going to take my sensor, I'm going to bring it as close to the brain as possible. I'm going to collect from a huge solid angle. I get high light collection efficiency. I get enough information to reconstruct a very large area. Now, why is that important for the brain? It's important for the brain because what I want to be able to do is sample from the largest possible area of the brain as I can and collect as much light because those photons are precious. Right? Each time I'm sending light into the brain and collecting it back, I have to work within a certain power budget and below a certain damage threshold.
This is very faint and... So I guess this brings up another question is like the sampling rate of this has to be really fast, right? Like cameras, they sample at like 25 hertz, 30 hertz, something like this.
Yeah.
But you might have to be going at like 1,000, right?
Well, it depends. So if we're using voltage sensitive fluorescent proteins, I would agree with you. Probably want to be in the 1,000 hertz range. But for calcium imaging, people are performing experiments using microscopes, using traditional sensors.
I've seen some of them. That's like in vivo.
Absolutely right.
It's quite amazing stuff.
And so those sampling rates are usually on the order somewhere between 30 and maybe 100 frames per second. And that's usually good enough for us to capture. At the 100 frames per second, we're looking at close to individual action potentials reported from calcium levels. Yeah,
I would say anything above the 100 frames, like you're gonna have to get special equipment. It's not off the shelf anymore.
Well,
maybe it is off the shelf, but
it is for scientific CMOS.
Okay.
So the cameras, basically the microscopes that people use to image in vivo neural data, those are off the shelf cameras, right? Sampling at 100 frames per second with very large sensors and very small pixel sizes. So those technologies exist, and so basically we're co-opting scientific CMOS, the sensors that people are developing for microscopy. We're throwing away the microscope body and replacing that with a combination of a mask to apply that transformation to the incident light, and an algorithm that can reconstruct that into an estimated scene.
Cool. And okay, so we've been talking about the theory. Have you actually built this, or what is it? What's been the experience so far with this?
Yeah, so we've built these based on off-the-shelf sensors. We've been able to image neurons inside of small animals. These are invertebrates like worms or jellyfish, things that we can easily manipulate in the lab. We've been able to see GFP, the green fluorescent protein. We've been able to image GCAMP, which is a common indicator of neural activity, and we've been able to reconstruct images with between 2 and 100 micron, depending on the conditions of the experiment. And that's around the spatial resolution that we would want if we were to look inside the brain. So our next steps now is to integrate that device with our illumination sources and implant that into the brain or lay them across the surface of the brain of rats and mice that have these genetically modified neurons. And that's probably something that we're going to be looking at doing over the next 3 or 4 months through a DARPA-funded program to develop these high channel density neural interfaces.
Wow. Amazing stuff. That's quite impressive. But I guess the problem with this would be that you couldn't do it in humans, right? Because you'd have to transfect. I mean, you'd have to basically infect a human, and that's not allowed, right?
I'm glad you brought this up. So one of the things that I learned at this meeting today is that there are multiple companies that are working on expressing optogenetic constructs in humans. And in fact, clinical trials are currently underway to treat blindness, where they are working to transfect the retina with channelrhodopsin.
Wow.
There's another company that's working on optogenetic deep brain stimulators, and they're planning clinical trials probably in the next year or two where it will transfect deep brain targets with optogenetic constructs. So I think these things seem like they're very far from clinical practice, when in reality, currently there are clinical trials for the retina, and I think in the next few years we're going to see stuff in the central nervous system.
Once you have the animal studies done, like that could be like, you could have the device ready, and they like right when they allow it, they're like, okay, everybody can do it, not just these few studies, and you're like, sweet, we have a device.
Yeah, and I think actually what's kind of interesting about this is that once there is a first proof of principle demonstration that shows safety and efficacy in humans, it might be retina, and then after that maybe central nervous system, that opens up many different applications, right? These techniques being genetic manipulation is already safe and effective. You can apply it for, say, deep brain stimulation, then it's easy to apply that to the other types of therapies that you would traditionally think about treating with electrical stimulation, but now with the optical specificity that you would give with the genetic.
Before, I don't know if we actually got this on the video, but you were talking about peripheral nerve. I mean, talking about the applications, like how it would just be the same thing, but just on the peripheral nerve, something like that.
Yeah.
And so the advantage of that would be, again, like specificity and all. Okay.
Yeah, so my understanding is that one of the challenges with the recording from the peripheral nerve is to try to understand where from within the nerve the signal is originating. Mm-hmm. And so if I were to put a cuff around the nerve, I can record the electrical activity, but I don't necessarily know with spatial resolution where within that nerve that signal is coming. Mm-hmm. So the idea is if we're using, if we have an optical approach, we might be able to do better specificity in terms of our recording. We also add that extra element, which is the genetically targeted aspect of it. Mm-hmm. So for instance, what you can do is perhaps with retrograde tracing, you can inject your transgenic into the end organ that will then transfect the cells that synapse onto that end organ. So you can basically retroactively trace all the nerves at a particular target because you've injected into the target that you're most interested in stimulating. And it'd be those nerves and only those nerves that would be activated by an optical approach.
And you could I guess split those up or filter that out and have like a red protein for pancreas or green one for liver or something like this, right? That's pretty clever, and especially like maybe that wouldn't be used, later on in clinical settings, but like definitely in the beginning, like to find everything, to map everything. Exactly. That sounds, it could be very, very useful. But this sounds like it's more for sensing, not stimulating, right? Or could you stimulate? How would you stimulate in this case?
Yeah, so we've been focusing mostly on the sensing aspect of it. Mm-hmm. Stimulation could be done potentially even with the same device, just by applying light and then your optogenetic constructs like channelrhodopsin, et cetera, channelrhodopsin for inhibition. You can imagine even having the same device operate both as stimulation and recording because we have to have illumination in order for us to record. That same technology that allows us to illuminate the source will also allow us to control the source. We had the complementary optogenetic construct.
Yeah, I mean, I've heard people talk about using different modalities to basically stimulate and to sense. So like you use optical for one and electrical for another, and ultrasound for another. Yeah. I'm not exactly sure, but I really like that because especially for the electrical stuff, like you kind of inject noise. I mean, like you really can't be stimulating and sensing at the same time. So this would allow you to do both. So it's really cool. Cool. So, what are your, I guess, next steps or something like this? What are your, you kind of talked about what you're working on the next few months. Like how many people are working on this? I mean, this sounds really cool.
Yeah. So this is a collaboration between several labs at Rice. In particular, one of my close collaborators, Ashok Raghavan, has a background in signal processing. So he thinks about how we can reconstruct images based on these complex measurements. Another colleague and close friend of mine, Caleb Kamari, is working on the in vivo preparations. So preparing rats and mice for these initial studies. I myself am working mostly on the systems integration and the post-processing of these sensors so that we can use them as these lensless imaging devices. So there's a group of three labs, many students. Jesse Adams, Vivek Bhumanathan were the lead authors on the recent paper that we had out in Science Advances. That was kind of the first demonstration of this lensless flat microscope. So it's a big team effort by a lot of people at Rice. And I think our next steps are to look at the animal models that I discussed, rats, mice, non-human primates, actually, something that we're trying to do within the year as part of the startup program. And this use in non-human primates would probably be the first step toward potential clinical application.
Yeah. Very cool. Very cool. This is very interesting stuff. Is there anything that I didn't ask that I should ask?
No. It was a perfect interview. You asked everything.
10 out of 10.
Yes, yeah.
10 out of 10 dentists agree. Okay, thank you.
Cool. All right, man. Thanks a bunch.
Yeah.